Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Alex Nicolaitis, the webcast coordinator for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we'll begin the Q&A portion, and I'll ask our speaker your questions. Your questions in the Q&A window will only be visible to myself and our speakers. So thank you for joining us today. It's now my pleasure to introduce our speaker, Linda Mathiasen, Strategic Customer Lead from Sativa.
Okay, thanks a lot for the introduction, Alex. I'm going to talk about how to develop big ideas in small-scale manufacturing of mRNA therapeutics, about how to overcome challenges in small scales, like need for flexibility and scalability, small-scale solutions to use for manufacturing, and handling the large amount of data that are generated from small but perhaps many parallel batches. But just to set the scene a little bit first, when we say RNA for use in therapeutics, it can mean a lot of different things. There are two major groups in the so-called RNA universe. The non-coding ones, they are shorter, and the production process starts with an oligosynthesis. These can, for example, have regulatory effects on gene level. Then there are the coding ones, the messenger RNAs. They are longer, larger, and the production process starts with an in vitro transcription synthesis from a DNA template. Sativa can support both processes, but my focus in this talk will be the mRNAs. In this group, we have variants like the self-amplifying RNAs, circular RNAs, and the more traditional linear mRNAs that we have seen quite a lot of the last couple of years. So mRNA has the potential to play a big role in preventing, treating, and even curing many of the most difficult diseases. Thanks to the rapid development of mRNA vaccines for COVID-19, the industry now has the momentum to overcome some of the challenges to really realize the true potential of this technology. There are some distinct benefits to the mRNA technology, such as speed of a cell-free process, the safety of a non-infectious virus-free vaccine, and the flexibility and cost of a process that can be scaled and standardized. This makes it promising for other infectious diseases like the flu, Zika virus, and potentially new pandemics, but absolutely also in the oncology space and for rare diseases, for example. If we look to applications, it has the potential to play a role in mRNA cell therapies, protein replacement applications, mRNA encoding for antibodies or parts of antibodies, gene editing applications such as CRISPR-Cas9 or zinc fingers, and as therapeutic oncology vaccines, to just mention a few. mRNA doesn't only span over a large number of potential applications, it also spans over a broad range of scales. From pandemic response, where you run a few large to mid-sized vaccine manufacturing batches per year for large patient populations and perhaps have localized manufacturing, to medium and small patient populations where there is a need for flexible manufacturing lines, perhaps multi-product facilities, and the possibility to scale out or up, and down to the truly individualized therapies, one drug to one patient, where we will have very many small batches of large numbers. So imagining manufacturing 10,000 to 100,000 batches to patients per year. And this is where we believe that we will have to see a lot of innovation happening. We see already today that workflows being collapsed and integrated to microfactories, and we see an increased interest in using non-plasmid-based DNA templates in the smaller scales to get rid of endotoxin challenges and to increase the speed. So are there different requirements for the different scales when it comes to manufacturing? Well, looking at the large to mid-sized batches, for example, vaccine manufacturing, needs are really there for high yields, high quality, high capacity, and you want to have a big throughput in your facility. In the smaller and even personalized batches, these parameters are still important, of course, but it is a great need for integration, for connecting the unit operations, and for automation, both when it comes to the run of thousands of batches and taking care of the data and perform batch release. As mentioned, a scale-out strategy is required, for example, for personalized scale. Cost of goods is important because you will have fewer doses to take the cost of manufacturing. Samples will go to QC, and patient tracking starts already when a biopsy from the patient is taken, to generating the right sequence through manufacturing and back to the patient. So as mentioned, we expect to see a lot of innovation in this scale that will probably also influence larger scale manufacturing in the future. There are different ways of handling the process diversity in smaller scales, each with different drivers. Either you scale down your separate unit operations and connect them physically with tubing as well as digitally, or you can go for a fully integrated approach, like an mRNA printer, a box concept. In this case, you might gain throughput and maybe cost of goods in some cases, but you will have to compromise on flexibility to adapt to different IVT and purification strategies. With connecting unit operations, you will have the flexibility to adapt your manufacturing line to accommodate for different types of linear mRNAs, but also for the other variants like self-amplifying and circular RNAs. So when you are in these smaller scales, what output can you get per year from a line? Here is a calculation example with a 2, 5, and 10 liter line with the assumption of a titer of 5 grams per liter and 46% overall yield. As you see, you can get a decent output from a 2 liter line in the blue here, around 200 grams per year. Even smaller scales can be supporting different phases of a project, late screening, process development, clinical batches, and perhaps even manufacturing scale if small patient populations or personalized therapies. When the size of the process goes down, smaller volumes are processed and the runtime will be much lower. So you will be able to fit in a much larger number of batches in a year in your facility. And actually a ballroom approach could be an option here. But then you really need to be consider about having closed systems and single-use flow paths so that you reduce cleaning between batches and products, that you prevent carryover and to increase speed. So in this overview, you can see how scalable single-use equipment can be in the whole mRNA workflow, including both drug substance and drug product. Earlier, we pushed the limits up to larger batches, but now we are pushing the limits down to support smaller processes as well. You can see that one system can span a large range of volumes just by changing the flow path. We also have a non-single-use equipment for process development, as you see in the lower part of the picture. And we also have launched actually a single-use chromatography system a couple of weeks ago, ECTA-ready 450. And it is not visible in this overview, but it will enable you to go even lower in scale. I will come back to that soon. But first, I would like us to look into the flexible IVT reactor that can take from 150 milliliters up to 25 liters. And what this data shows is that we can scale an IVT reaction from 1 to 10 milliliter in test tubes and to 150 milliliter in a rocking bioreactor and get consistent output. It is also important to have control of the RNase activity. Many single-use devices don't have RNase-free certificates, but we can see in the lower graph here that the bag we use coming out of manufacturing and is gamma irradiated shows very low levels of RNase activity. It is comparable with the negative controls. And it is really a key to control your RNase activity in your lab and in your facility. And now a bit more about the newly launched chromatography system, developed for use in small-scale manufacturing of mRNAs, but also for other modalities like viral vectors or monoclonal antibodies. It is called ActiveReady 450, and it has a low hold-up volume and low flow rates to support small-scale purification, and it is a closed single-use system to reduce contamination risk. And it's also designed to have a benchtop footprint to enable compact processes in your facility. With intensification, with smaller batches, but a larger number of processes, comes a strong need for digitalization. Imagine having multiple lines in parallel, as mentioned earlier, maybe thousands of individualized batches per year, and you will have to release them, take care of all the data coming out from the unit operations. So data management will definitely increase. Also, keeping track of information connected to one patient from biopsy through manufacturing and back to the patient will be a key for personalized therapies. There are often many manual entries or transcriptions that needs for eye verification. And also one batch can actually have up to 200 consumables that needs to be logged in a batch record. And if this is done manually, it takes a lot of time and effort. Digitalization will be of great importance as the scale decreases and number of batches or programs per year increases. Also, FDA has a driver towards electronic submissions. So last but not least, digitalization can start with connecting your unit operations and let data automatically go to a secure place in a historian. It can then also be connected to other systems like MES, LIMS, and other digital solutions that support quick batch release. You are gaining process data analytics, regulatory compliance, and reporting support as well, going in this direction. And now I come to the very end of this talk, and I would like to summarize a bit. I truly believe that big ideas can become true in small scales when it comes to mRNAs. A couple of key takeaways are, so keep manufacturing in mind early, in all stages and scales, to save time and money if you need to go back. And stay flexible with equipment platforms and standardization. Flexibility will give you the possibility to quickly adapt to different patient population sizes, dose variations, and process diversity, and to accommodate mRNA with different characteristics, linear, self-amplifying, circular, and what will come in the future. So flexibility will be a key in the rapidly moving field of mRNA. And with that, I would like to thank you so much for listening, and now I'm happy to take any questions.
Okay, great. As a reminder to our audience, you can go ahead and type in your questions for our speaker now. So our first question is, you mentioned that you think that innovation will happen in the smaller scales. Can you elaborate a bit on that?
When it comes to small scales, I believe that intensification and integration of the workflow is a key, but still keeping the flexibility to adapt to different products and different characteristics of the drug substance and product. That will be the key. So automation, digitalization, integration, it will be more urgent. And also for the personalized cancer vaccine, for example, patient tracking and traceability and QC and automated process control. I think that all of these things will be of great importance and will encourage to innovations to make the processes quicker and even more robust. When waiting for Alex, if we look at the different unit operations, I believe I mentioned in the talk that the increased interest in non-cell-based DNA template creation, to get rid of endotoxin challenges and increase the speed. But I also believe, if you look at the IVT or the in vitro transcription step, that is another step where I believe that it will be a lot of innovation happening, because the raw material, the enzymes, they are so expensive, and today the reaction is mainly done in batch, so perhaps alternate reaction designs sort of that reduce inventory of expensive raw materials could sort of make a difference. I also believe that if you look at the purification steps sort of downstream of the IVT reaction, that is an area of improvement too, because mRNA, as we have seen, mRNA don't interact well with sort of the traditional chromatography resins. For example, I mean, the mRNAs are bigger, they have sort of another impurity profile, so I believe that sort of a toolbox to mix and match from, sort of to adapt to the different mRNA variants, could be of great help. But hopefully downstream steps can be removed if we get sort of technical solutions upstream that are fit for purpose for mRNA. So there are many areas where innovation can happen.
So the next question is, does gamma irradiated items can be used regardless of an RNase free claim?
I mean, so at least what we have seen sort of in the work that we are doing, we don't have these claims, but then we have done these studies as I referred to that the R&D team has been performing, looking into how it looks, and that the sort of the RNase levels are really sort of very low coming out from there. So not saying that it is sort of if it's connected or not, but that is at least what we have seen, even though we don't have the strict claims.
Now, are you seeing the same scaling efforts in the DP space?
So DP, I interpret that sort of in the drug product, where you have so the encapsulation piece and the downstream steps. I don't know really what scaling you sort of refer to, but they will also definitely go down in scale and follow the sort of the size of the total process. But there are quite big scale differences if you look at sort of drug substance and compare it with drug product. Because going into the LNP formation, for example, it's at least a 16-time dilution happening there. So the sort of the downstream steps or the drug product steps will be larger in scale, but still relative to the manufacturing size. I hope that answered the question.
And what is the capacity in terms of G of RNA per M2 dose of membrane that you propose during the process development? Excuse me.
The membrane, I guess you refer to the TFF piece. It is a little bit dependent on where you are sort of in the process. If you are sort of in the mRNA drug substance manufacturing, or if you are sort of in the downstream. And I think that's sort of where you need to look, dependent on how your product looks, how large it is, sort of what are the characteristics, and scale that sort of after it's sort of or adjust it and optimize it after the scale that you are in. So I can't really give a straight answer to exactly what to use there. We have to look into sort of the whole process.
Okay, so it looks like we're running short on time. If we don't get to your question, we'll be passing them along to Linda, who can follow up with you directly. So please go ahead and continue to type in any questions you have, and we'll get those passed along. The last question for the webcast is, do you think the need for single-use components will continue to grow as the solutions move towards smaller batch size, higher batch run number?
Yes, I do. I think that is one of the main sort of benefits. So if you're working in many parallel processes, because you can have the process closed, and you will have quick turnaround times or turnover times between, if it is sort of between patients or between the smaller batches. So yes, and that will also put a bigger pressure on the sort of security of supply or the supply chain things. So I understand that is sort of a big thing that we need to work on. But I definitely think that it will be a lot of benefits going single use when you go to smaller scale, and that you have a diverse sort of biological pipeline. Yep.
All right. Well, thank you, Linda.
Thank you so much, and thanks for really good questions. And don't hesitate to reach out if you have any other questions or thoughts. We'll be happy to answer.
And thank you to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.